course, eukaryotic cells are going to undergo RNA processing after the RNA is transcribed. And what that involves is just a couple of things. First of all, non-coding segments called introns must be removed. Introns are segments that are not involved in the production of the particular protein that we are trying to accomplish today. They might be used for some other purpose, but they're not going to be involved in this protein. Also, we have to add a 5' cap and the poly A tail. This allows the mRNA transcript to leave the nucleus. Now, we've already got the polyadenylation signal sequence, that's several A's, 1035 A's at the end, but we're going to be adding 50 to 250 more A's on the end. And that's what this is right here. Just, 50 to 250 more adenine residues. All right, a 5' cap is actually a modified GTP. And then there is a little section here called the 5' UTR, the untranslated region. Here is a start codon, and hopefully some of you know that's AUG. And we have our protein coding segments that are our exons. There's an exon here. I think I should have written start here, actually. Let's correct that real quick. Start. So sorry. That's really messy. And then another protein coding segment here and a segment here. These are called exons. And they can be numbered, and when you start studying genetics, they'll be like, exon 6 involves a mutation at blah, blah, blah. That's what they're talking about. So here's my exon, exon, exon. The introns have been removed. When they are removed, this is called splicing. So how does splicing work? Splicing is accomplished by something called the spliceosome. Whoever came up with the naming conventions for these things was really not very creative, but that's all right. So spliceosomes are responsible for splicing, and really what's happening here is that the spliceosome is incorporating several other kinds of RNAs. We call them small nuclear RNAs. Those come together with other proteins along with the pre-mRNA, the immature mRNA. That should be a little m. And then those small nuclear RNAs bind to certain regions and cause the two ends of introns to come together and loop, and then they would just be cut right out. And what's left is our mature mRNA. So interestingly, because of this, we sometimes talk about RNAs functioning as an enzyme. And this is a really interesting section of biotechnology that we could spend hours talking about. There are 3 properties of RNA that can enable it to function as an enzyme. It can form a 3-dimensional structure because it can base pair with itself. And that's really interesting. Sometimes RNAs contain functional groups that can participate in catalyzing reactions. Hopefully you remember what functional groups are. They're little additions of certain things like hydroxyl, carboxyl, carbonyl. Remember those names? So this functional group sometimes can act as catalysts for reactions, can participate in catalysis. And lastly, sometimes RNA may hydrogen bond with other nucleic acids. I ran out of room here, sorry. So RNA may hydrogen bond with other nucleic acids. And there are many RNAs that we have not talked about, different types of RNAs that, they're discovering new RNAs all the time, it seems like, and they have all kinds of interesting functions that we're just not really sure we know a whole lot about.